Hi, my name is Tim. For those that don't know me, I'm a biology professor at Mount San Antonio College. And today we are going to walk you through Lab 13, the Respiratory and Circulatory System. And I want to point out that this is a general biology teaching purpose video and is not meant in any way to be designed for a professional medical advice of any way. You should always talk to a doctor, a physician, if you have any medical questions. This is just for teaching purposes. Once again, my name is Tim Ravel. I'm a biology professor, and my colleague and I, Xavier Lopez, who's also a good friend and colleague and biology professor, will be walking you through today's Lab 13. Now, before we go too far, I'd like you to pause the video and get your lab book out. And before we go any further, I'd like you to familiarize yourself with some of the terms that are in the beginning of lab. So the first 2 or 3 pages of lab explain some terminology, and it would be useful for you to read those before we go any further. So pause the video now. So let me walk you through kind of what's happening here, so you have an idea of what we're going to talk about today. So we'll start, in my case, about talking about the respiratory system. And what's going to happen is, through your nose or your mouth or both, you can draw in air, and that air can come in to your nose or mouth, and it goes through a very large tube that you have called the trachea. And the trachea is right here, right by your throat, and the trachea travels down, and then it branches into what are called your bronchi. A bronchus is singular and plural. Bronchi is more than one. And then those branches continue to happen and get smaller and smaller and smaller, all the way down to the very end at a microscopic level. You have these little sacs, these microscopic sacs, and those are called the alveoli. Once again, that's a plural, and alveolus is a singular one. And they're like grape-like clusters at the ends of your lungs, and your lung is mainly these little pockets, these spaces, and they're very important because that's where the air exchange is going to occur. Now, in the case of an animal such as yourself, as a human, we are what are called negative pressure breathers. So the way we get air in our lungs is by creating negative pressure. So the diaphragm, which sits right here, connected to the bottom of the lungs, will contract, and it will move down, kind of like what you're seeing here in this video. When it contracts, it pulls and it stretches the lungs, and that makes the space in the lungs larger. And so the outside air, which is now at higher pressure because you've reduced the pressure in your lungs, it draws the air in. And then going around the alveoli, wrapping around them, not only is there a blood supply coming to it and from it to exchange gases, but there are these fibers that are kind of stretchy. And so when you pull them or stretch them, when you relax the diaphragm, they rebound, they constrict, and they help push the air back out, okay, during normal breathing. Okay, now, right now as we sit here talking, we are undergoing, hopefully, unless something unusual is happening, we're undergoing normal breathing, which is called eupnea. Now I'm talking, so that sort of throws off my eupnea, but if I were just sitting, relaxing, and not talking, I'd be breathing in and out at a normal pace and a normal breath. So normal breathing, we call that eupnea, normal breathing. And in the scenario of eupnea, I am breathing in and out a specific volume of air, and we call that volume of air a tidal volume. And it tends to be around 500 milliliters of air. Okay. Now, as you're sitting there thinking about this now and you're breathing in and breathing out, you probably know that you could, if you chose to do so, you could breathe in more air than you're currently breathing in. So the air beyond that tidal volume is your inspiratory reserve volume. That's the extra amount you could breathe in if you chose to do so. And that's important because that allows us to do things like exercise, because as we start to exercise, we have the ability to breathe faster and deeper, which allows us to continue to exercise beyond that tidal volume. We can also, you'll notice as you're breathing in and out, you could also, if you wanted to, push out more air than you wanted to. So that extra amount of air I could push out is my expiratory reserve volume. And then if I put those two together, or the three together, my inspiratory reserve volume, my tidal volume, and my expiratory reserve volume, collectively, we call that the vital capacity, which is the sort of the maximum amount of air you could breathe in and push out under your own power. Now, even when you breathe out all the air you can, your respiratory system is still a hollow tube. They don't collapse completely flat. So there's a little bit of air still left in there, and we call that the residual volume, or sometimes I think in your lab book they call it the dead space or the anatomical dead space. So there's still some air inside there, and we'll get to that when we're talking about that in a minute. These volumes of air, and you might have seen one of these devices before. They use these often in hospitals to help strengthen your lungs if you had pneumonia or surgery or that type of thing. And the movement of the balls here in this particular, there's different versions of this, but moving those balls corresponds to different volumes of air you're moving. And in Bio 1, we use our respirometer like this one that I'm going to show you here to measure both tidal volumes and our maximum exhalation volumes. And so I did this, and in my case, I find that I have a tidal volume, typically somewhere around 500 to 600 milliliters of air, and then I have a maximum exhalation volume of around 6,000. Sometimes I get up to 7,000 milliliters of air in a maximum exhalation breath. And then my respiratory rate tends to sit somewhere around 16 breaths per minute. You can measure your own by trying to count how many breaths you take in a minute by timing it. It's a little tricky to do that yourself, because as you think about your breathing, it starts to sort of influence how you breathe. So often it's better to have somebody else count your breaths for you, or to have a device is more accurate. Now, if I use that data, 16 breaths per minute, I can calculate my pulmonary ventilation rate, which is about 9,600 milliliters of air per minute, and my alveolar ventilation rate, and that's where you take that dead space out and you remove it. That's around 6,400 milliliters of air per minute. So on page 239, if you want, you can use my data that I'm providing in this video that I'll put up on the screen, my calculations, or you can use the sample calculations they provide in lab. And if you want, you could actually make your own homemade respirometer. Again, not a medical device by any means, but just for Bio 1 teaching purposes. So I'll put a link up for how you can make your own respirometer if you want to see kind of how much air you can push out of your lungs. You can use that if you choose to. Okay, you might notice that in my calculation, I use a different value for dead space, and that is due to an estimate on my part that my lung size is probably a little bit larger than the average person. It's sort of based somewhat on body weight, and my body weight tends to be slightly higher. So I use a value, instead of 150 milliliters of air, I'm using around 200 in my calculation. But you can feel free to adjust that how you want. Now, for this part of the experiment, I went over to my in-laws' house on Mother's Day, and I recorded myself holding my breath in the swimming pool. And in this first video, I'm holding my breath after a bout of eupnea. So my eupnea, again, is normal breathing. And then I measured how long I could hold my breath in that scenario. And then I also forced myself to breathe and to hyperventilate. So I was breathing more rapidly and deeper than I needed to. And I did that for about a minute of hyperventilation. And then I compared the two. And as you can see, during eupnea, my normal breathing, I held my breath for about 38 seconds. But after I hyperventilated, I was able to hold my breath for about a minute and 9 seconds. So why the big difference? Why almost twice as long? Why do you think that might be? What most people guess, which is I think it's a reasonable assumption, is what people think is that when I hyperventilate, I'm able to get more oxygen into my lungs or into my blood, and therefore I can hold my breath longer because I've got more oxygen. That's sort of the typical answer that most people come up with quickly, which is really not correct. So I'm going to explain that in a minute. Now, one of the ways you can sort of see that is if you have one of these oxygen sensors that you put on your end of your fingertip, where it measures your oxygen saturation. Most of you will have, as long as you're, fine and healthy and whatnot, will be around 97, 98%. And if you hold your breath, that value will drop as you're holding your breath, because there'll be less oxygen that's reaching your fingertip. But you'll find that it doesn't take more than just a couple of breaths normally to get back to around 100%. So my point is that if you were to hyperventilate, you would go from normal breathing and you would hyperventilate, and maybe your oxygen meter would only go up 1 or 2%. So it's really not changing that much. You're almost at 100% during eupnea anyway. I should also mention that you can see this is just a sort of a depiction of different oxygen meters you can buy. These are probably not medical grade oxygen meters. These are also, again, for the purposes of like a Bio1 lab for educational or home purposes. The more expensive and accurate equipment is probably not $20 or $30. But anyway, my point is that if you have one of these around, you might be able to see that as long as you are normal, healthy, lung functioning scenario, that you're probably around 100% anyway. Okay. Now, in this experiment from your lab book, also, just to sort of give you a hint at maybe why that is, we have 3 test tubes. And one of the test tubes has hydrochloric acid, HCl. One has sodium hydroxide, and one has water. And this is on page 238 of your lab book if you want to follow along with this experiment here. And so what we did is we also added to all 3 tubes an acid-base indicator called phenol red, which will interact with the solutions. And the acid solution you see turns sort of a yellow or orange color, while the other 2 tubes stay sort of a dark pink or purple color. You can pick what you think the colors are. I'm not very good with colors. To me, there's 12 kinds of colors that I'm pretty good at. There's green, there's blue, there's so if my color description doesn't match what you see, you're probably right, but it'll still make sense, so follow along. Okay, now you're going to watch this video, and what's going to happen is I'm going to gently blow with a straw into the solution with the water, and watch what happens when I blow into the one with the water. You'll notice that what's happening is two important products are coming out. When I blow into the tube, I'm breathing out CO2, and that carbon dioxide goes into the tube with the water in it, and it reacts with the hydrogen in the water, and it makes that tube acidic. It makes it like the one with HCl or hydrochloric acid in it, the CO2. That part's important. You'll also notice on this piece of glass that I'm going to breathe on here that another product comes out, which is water vapor. So two things are coming out of my breath, at least, that are significant. One is the water vapor on the glass that you can see, and the other is the carbon dioxide. And again, the carbon dioxide comes out and it interacts with the hydrogen in the water, H2O, and it produces an acidic solution, which is why it turns yellow and it looks sort of like the acid base. So this comes back then again to, why can I hold my breath longer during hyperventilation? I'll give you another hint. It has far more to do with the carbon dioxide and the acidity of your blood than it does with the oxygen. Okay, so I hope this video helps you get started on lab number 13, and good luck as we move forward, and let's see what we can learn next.